Hello, my name is Sandra Almanza. I am a junior in anthropology pursuing a career in primate conservation. My drive behind the career that I've chosen are the rapidly depleting numbers of primates that are left in the wild. They are being affected by deforestation, the illegal pet trade, and bushmeat hunting. My goal is to use conservation genetics as a tool to help with maintaining the levels of biodiversity. My SURF project was about establishing a molecular primatology research pipeline. Now, have you ever wondered what it takes to go from the field to the lab? Allow me to walk you through these steps. Our field researchers must first complete and submit compliancy forms at an institutional, national, and international level. These organizations may include, but are not limited to, the FAU Division of Research, IACOC, CITES, and the CDC. Now, after completing the compliancy component, we can move on to the next step, the biorepository. Our lab checks and receives the source material packaging from Africa. We check the tubes for proper field identification, cracks, or adding extra preservative if needed. We transfer the source material into new 8 milliliter tubes that go into long-term preservation and storage in our lab's -80-degree freezer. We apply a unique lab identifier and enter the information into the biorepository database. This leads me to my next step, the molecular genetics bench work. In this step, DNA is extracted from the fecal or tissue samples. We then amplify the DNA target region and visualize the product by using gel electrophoresis. We then send the product off for sequencing. And this leads me to my final step, bioinformatics. We receive these sequences and we check them for accurate base calls. And we bring them into an alignment program in which we add more sequences in order to conduct an accurate comparison of phylogenetic relationships. Now, after adding them to the alignment program, we run statistical analysis to acquire the posterior probability and bootstrap values. To visualize the output from the phylogenetic programs, we bring them into a graphical viewing program for the purpose of producing publication-ready figures. Now, the purpose of this lab was to establish a molecular primatology research pipeline, and we hope to apply these steps as well as to apply my new skills in bioinformatics and molecular genetics to an independent research project on the origin of the Dania Beach vervet monkeys. Thank you very much.